Hi everyone, I'm Marius. I'm a PhD student in the lab of Fabian Theis at Helmholtz Munich, and I'm happy to present moscot-lineage today, which is a tool to map lineage-traced single cells across time points. Before I actually start, I want to highlight everyone that's involved in this project. This project is co-led between Zoe and myself. Zoe is a PhD student in the lab of Mor Nitzan at the Hebrew University in Jerusalem, where I was on a lab exchange this year in the summer. And also Michael from Fabian's lab, and of course Fabian are involved in this project. Okay, so let me start with giving an overview of my talk. There are three parts. The first one is a motivation. Why do we need this type of model? In the second part, I introduce the model. It's called moscot-lineage. And in the last part, in the third part of this talk, I give two applications. So let's start with a motivation. In an ideal world, we would be able to sequence a population of cells at different time points, and we would be able to sequence the same cell at different time points. Some cells will proliferate, other cells will die. And in the end, since we have sequenced the same cells, we would be able to say something about differences among early cells, among progenitors, that then give rise to different fates, and we could study these differences and build hypotheses about what it may be that gives rise to these different fate decisions. In the real world, however, we don't have these links because we can only sequence a cell once. And in many time series experiments, these different time points actually correspond to different individuals, to different animals, which means you have to probabilistically reconstruct this mapping between the different time points to be able to study fate decisions in a system like this. And in the past, the method that has been successfully applied to this is optimal transport. So let me give you a brief introduction to optimal transport. I'm going to use a very simple example from the Python optimal transport toolbox here, which is about transporting bagels in Manhattan from bakeries to cafes. So in this map here, we have red dots for the bakeries, and the size of the dot corresponds to how many bagels they produce. And the blue dots would be cafes. And again, the size of the blue dots corresponds to how many bagels each cafe needs. And the problem is obviously all the bakeries want to ship all the bagels to the cafes in such a way that the overall distance or the overall cost associated with this transport is minimized. So to formalize this, we can write down a cost matrix, which is some distance between bakeries and cafes. For example, Euclidean distance, Manhattan distance, whatever. And we label the different bakeries and the different cafes like that. And here you can see, for example, the distance between bakery 0 and cafe 4 would be quite high, because 0, that's the red one here, which is in Hell's Kitchen, and 4 is down here. So there's a very large distance, which is why the cost to transport a bagel from here to there is quite large. So we have the cost matrix. What we want is a coupling. The coupling will tell us how many bagels go from here to there. And we have marginals. The marginal mu on the left side tells us how many bagels does each bakery produce. And mu tells us how many bagels does each cafe want. We can formalize this in an optimal transport problem, which just says we want to get the minimum over all the entries in our coupling, so how many bagels go from here to there, multiplied by the associated cost. We sum over all the different entries in the coupling, and we want to minimize that, subject to these constraints that basically ensure that all bakeries get rid of all the bagels and all cafes get all the bagels they need. That's just what these constraints say. So you can do that, and you get a solution like this, which is sparse. Usually every bakery only ships to 1 or 2 cafes. And this is very nice. It gives you a solution to the problem. The limitation of that is that computationally, that is a little bit challenging. It's computationally quite expensive to compute this. So what people often solve instead is the so-called regularized optimal transport problem, where you add this entropy regularization term here. So h of pi would be the entropy of this coupling pi. And what it means in practice is that there's some entropy introduced into this transport matrix or coupling matrix. So now every bakery delivers to more cafes than before, and the whole thing looks a little bit more smooth. And it's computationally much more efficient to solve this problem. Okay, so that's optimal transport. This has been applied in the single cell field before very successfully. I'm highlighting an example here from Jeffrey Schiebinger from 2019, where they studied mouse embryonic fibroblast reprogramming to different endpoints here, and they did exactly what I described before. They basically used optimal transport to link the distribution of cells at different time points in a time course study with very high temporal resolution. So they sequence very often. And basically, once you have inferred your couplings that link the different time points, you can answer many questions that are interesting from a biological point of view. You can say something about descendant distribution. So this cluster, where does it go in the next time points? Same for ancestor distribution, also for something like shared ancestry. So given two populations that are different at some time point, when did they diverge? So when was this fate decision probably made? So this works really well in this example. However, the limitations are that when you go to larger time gaps, it does not work very well any longer, because when the gene expression distance becomes too large, it becomes really ambiguous which cell from time point T1 you should match to which cell from time point T2. And the other limitation is that if they're hidden variables, so say the lineage priming at the earliest time point is actually not established on the gene expression layer, but potentially on an epigenetic layer, then this is not contained in the data that you sequence in a single-cell RNA-seq experiment. And so you may not be able to make this fate prediction very well based on just linking gene expression profiles together. And this has also been shown before here in this paper by Caleb Weinreb from Alone Klein's lab. So one solution to get around these limitations, to overcome these limitations, is to include additional priors that regularize these mappings. And the prior that we are interested in this project is lineage tracing information. And lineage tracing is like a very large field, and there are lots of different approaches to lineage tracing. What we are interested in here are approaches that give both state readout at the single cell level, so typically gene expression, and a lineage tree. So we only consider approaches that give us both. In single cell lineage tracing, there are prospective approaches where you engineer your barcodes into the cell, and there are retrospective approaches where you just make use of naturally occurring barcodes, so mutations. For prospective, these can be static, so the barcodes don't change once you have introduced them, or they can be dynamic. So barcodes accumulate mutations as development goes on, so you get increasing barcode complexity. And on the retrospective side, this can be done either in the nuclear DNA, or it can be done in mitochondrial DNA. And I'm just highlighting some methods here, but this is not comprehensive. There are many more. There are orange boxes around dynamic barcoding and mitochondrial DNA, because these are the two settings we are targeting in this project. Now, I want to go a little bit more in depth for the dynamic barcoding. These approaches usually work by introducing a barcode into the DNA together with a CRISPR-Cas editing system, which continuously edits the barcode, introduces indels. These indels are inherited to daughter cells, which continue to accumulate mutations. And in the end, you get something like this. So you get, for every single cell, you get this mutated barcode with different indels. And from there, you can reconstruct a lineage tree. This is true for all of the methods here over on the left, except for tracer-seq, which works a little bit different. And I'm not highlighting mitochondrial lineage tracing here because we don't have an application example for that in this talk, but the method works also for this setting. Okay, so now let's go into the concept of this a bit more. So say we have two individuals. In this case, it's just a single cell, a fertilized egg. And this cell is barcoded with a dynamic barcoding system. So the barcode here just consists of 3 different colors. Now I let this cell divide, differentiate, grow, and so on. And it accumulates mutations because I have this dynamic barcoding system in there. And in the end, at time point T1, I sequence my cells. Some cells I will observe and other cells I will not observe. Now for individual 2, I also start with a barcoded cell. I wait a bit longer, I accumulate more mutations. And in the end, at time point T2, which is later than T1, I also sequence some cells and other cells I don't sequence. So what I get from this as a readout basically is barcodes for both individuals, as well as gene expression. Now, to link these two together, It's easy to do that on the gene expression level. For that, we can just use the Waddington OT model from Jeffrey Schiebinger that I introduced a few slides before. However, you cannot do that on the barcode level because the barcodes have been generated randomly in each individual. So you can use barcodes within one individual to reconstruct the lineage tree. However, you cannot compare them across two different individuals or across two time points in this setting because they don't have anything to do with each other. However, we still want to make use of them in our model. So let me introduce the model in the next step. Before I do that, there's one other flavor of optimal transport that I have to introduce, which is called Gromov-Wasserstein optimal transport. And it's easiest to explain that with an example. And this example comes from a paper from Mornitsan, where they were interested in mapping cells from gene expression space to physical space. So given just gene expression profiles, what's the likely arrangement of these cells in my physical space? And the way you can do that is with this Gromov-Wasserstein optimal transport. What you do is you say you have two metric spaces. So this metric space X would be gene expression in this case. Y would be your tissue space. So it would be Euclidean space usually. And you say you can only measure distances within each respective space. So you have your distance matrix Cx, which lives in X space, and you have distance matrix Cy that lives in Y space. And what you can do, you can make pairwise comparisons. So you can basically say, if I and J are similar in space X, they should be mapped to a pair, K and L, that are similar in space Y. So whereas in standard optimal transport, you always make one-to-one comparisons. You say, how similar is this object here to that object there? In Gromov-Wasserstein optimal transport, you always make pairwise comparisons. You consider two pairs here in this space, you consider two pairs in that space, and you try to find a mapping, a coupling pi, that enforces concordance of distances. So on average, across cells, across genes, cells which are similar in gene expression space are going to be similar in your tissue space. This doesn't hold for every cell and for every gene, but on average it holds. And that is the objective function you get from that. So you have a distance between these two distance matrices here. You multiply this with the respective entries in the couplings, and again, you use an entropy regularizer. So let me recap what we have. We have two time points. We have gene expression from both time points, as well as barcodes and marginals. And these marginals basically could reflect uncertainty we have about cells or just any weighting. If we don't know anything about them, we just put them to uniform. What we want as an output is this coupling matrix pi that tells us which cell from the earlier time point, they would be in the rows here, is linked to which cell in the later time point T2. The way we do it is by using a Gromov-Wasserstein setting. So we have two metric spaces corresponding to T1 and T2 in barcode space. We compare barcodes only within one time point. So here in time point T1, we can say this barcode from cell I is similar to this barcode from cell J. And we do the same thing in time point T2. Additionally, we also have gene expression information for all of the cells. And in this gene expression space, we can make one-to-one comparisons across time points between cells. The final objective function is basically an interpolation between these two terms. It's called Fused Gromov-Wasserstein. So you have a Gromov-Wasserstein term here. That's the first one here. That's weighted by alpha, which is the one I introduced on the previous slide for Moore's model. And the second term here is a Wasserstein term. That is basically what we saw before for the bagels. That is a standard optimal transport term where you link cells directly across time points. And we have an entropy regularizer. And there's this weighting term alpha that basically determines how much weight do we give to Gromov-Wasserstein, how much weight do we give to Wasserstein. Once you have this coupling, you can feed it, for example, into cell rank to get out state trajectories, so fate probabilities, and putative driver genes. Right. Now I'm going to go into two applications. Before I do so, I'm going to introduce the only competing approach here that works in this setting, which is LineageOT from Aidan and Jeffrey. The aim here is the same. They also want to map between two observed time points. They construct lineage trees from barcodes at the last time point. they used the last time point, the lineage tree, to pool together cells that are related in lineage history. So these are like inferred ancestral states, and then they link these inferred ancestral states to the earlier time point by using a standard optimal transport approach. So no Gromov-Wasserstein approach, just a standard optimal transport approach to link them. So we adapted their simulations from their paper, where they look at 4 different trajectory topologies: bifurcation, convergent, partial convergent, and mismatched clusters. And we consider 2 different settings. One is the setting where we give the true tree to the algorithm, because this is simulated data, so we know the actual ground truth lineage tree. And the other setting is where we use a fitted tree. So we use a reconstruction algorithm like Cassiopeia or neighborhood joining to reconstruct the tree given the barcodes. We compare 4 different methods. One is optimal transport. The other one is Gromov-Wasserstein in green. Then you have moscot-lineage in red, which, as I said before, basically interpolates between the optimal transport and the Gromov-Wasserstein term and borrows information from both. It borrows information from lineage space, and it also borrows information from gene expression space. And we benchmark this against the LineageOT model. Okay, here you have on the y-axis the mean error. So we compute 2 errors. We compute something we call a ancestor prediction error and a descendant prediction error. And then we take the average of that, and that's what we plot here as the mean error. And we do this for all the different trajectory topologies for both settings. For the true tree, we find that usually either moscot-lineage or Gromov-Wasserstein have the lowest mean error. So they are the best competing methods here. However, when we go to the fitted tree, Gromov-Wasserstein becomes much worse than moscot-lineage. So moscot-lineage retains its really good performance when we go to the fitted tree. However, Gromov-Wasserstein relies on the tree a lot. It gets no other information. So if there are errors that come from the fitting procedure, then the coupling is just going to be very wrong. And moscot-lineage can correct for this by also using gene expression information, and is consistently the best performing method in terms of this error metric across all trajectory topologies considered here for the fitted tree. We then ran to another example to real data to see elegans development, where the ground truth lineage tree is known. So in this study here, Tucker et al., Science 2019, they used the ground truth lineage tree for C. elegans to basically annotate each cell with a node from this lineage tree. And for a subset of cells that looks like this, so there's a certain nomenclature how you call these lineages. And that's basically what we used as the lineage tree that we fed into our method and also into the other methods. So here there are different time points, and we did this separately for each pair of time points. So this would be the 170 minutes to 210 minutes time points that we linked together, and so on. And again, we always computed the mean error, and we see that moscot-lineage is either the best performing method or it's the second best performing method after Gromov-Wasserstein. But in some situations, Gromov-Wasserstein has a really high error because something went wrong in the tree reconstruction. And again, moscot-lineage is much more robust to that. And it also outcompetes the other methods here in terms of this error metric, optimal transport and LineageOT. We then zoomed into one of these time points, 210 to 270 time point. We computed the difference in ancestor error. So if it's red here, it means LineageOT has a higher error than moscot-lineage. And there was one cluster where the difference was especially large. So we zoomed in on that, looked into the ground truth coupling that we get from the ground truth lineage tree, which told us that this entire cluster should basically be coupled to this one cell. We then checked what LineageOT did. It coupled this cluster to many, many different cells, whereas moscot-lineage coupled it exactly to that one cell, basically by using barcode information from both time points and by combining this with the gene expression information. We next took a more global perspective and computed couplings for all the different time points and composed them together to basically get a global coupling from this 170 to this 450 time point. We focused on one cluster here, this GRL cluster. We computed the coupling matrix, put this into cell rank, got fate probabilities for this GLR cluster, and then looked for putative decision driver genes using cell rank based on our coupling matrix. And we found these 2, which are also implicated in this process in the original publication. Right. that brings me to the end already. Again, I want to thank everyone involved in this project. Monique Zahn and her lab at the Hebrew Uni in Jerusalem, Fabian and his lab in Munich, and of course, Zoe and Mike, and you for your attention. Thanks a lot.